Dairy production has undergone a radical transformation in recent years, propelled by advancements in genetics, nutrition, and molecular biology. The science of nutrigenomics, which examines how nutrition influences gene expression, has emerged as a promising approach to optimize milk production and quality. By integrating genomics with precision nutrition, researchers aim to enhance lactation efficiency, improve milk composition, and promote overall herd health. This documentary delves into the molecular intricacies of nutrigenomics, highlighting the role of dietary modifications, genetic markers, and metabolic pathways in shaping milk yield and composition. Dairy farmers and researchers have long sought sustainable strategies to maximize milk production without compromising animal welfare. Traditional approaches relied on selective breeding and conventional dietary interventions. However, recent findings in nutrigenomics suggest that gene-diet interactions play a significant role in modulating lactation. By understanding the molecular pathways influenced by specific nutrients, scientists can tailor feeding strategies to activate beneficial gene expressions while suppressing negative metabolic effects. This approach enables the production of higher quality milk with enhanced nutritional properties such as improved fatty acid profiles, increased protein content, and optimized lactose synthesis. Milk production in dairy cattle is a highly complex biological process, controlled by numerous genetic and environmental factors. The advent of genomic technologies has significantly advanced our understanding of the specific genes and regulatory elements that contribute to lactation efficiency, milk yield, and milk composition. Recent research has identified multiple quantitative trait loci, single nucleotide polymorphisms, and genetic variants that influence these traits, offering unprecedented opportunities for precision breeding and nutrigenomic interventions. By leveraging genome-wide association studies, transcriptomics, and epigenetic profiling, scientists have been able to map the genetic architecture of milk production, leading to improvements in dairy herd management and productivity. One of the most well-characterized genes influencing milk production is diacylglycerol O-acyltransferase 1, or DGAT1, which plays a crucial role in triglyceride synthesis. Variants in this gene, particularly the K232A polymorphism, have been extensively studied for their effects on milk yield and fat content. Research published in the Journal of Dairy Science reported that the AA genotype of DGAT1 is associated with higher milk fat percentages, while the KK genotype correlates with increased milk yield but lower fat content. This trade-off presents a challenge for dairy producers aiming to optimize both milk volume and quality. In a large-scale study involving Holstein cows, researchers found that animals homozygous for the A allele produced milk with approximately 20% higher butterfat content compared to cows carrying the K allele. The functional impact of this polymorphism is attributed to an altered enzyme efficiency in catalyzing triglyceride formation in the mammary gland. As a result, selective breeding programs now incorporate DGAT1 genotyping to tailor milk production traits according to market demands and nutritional goals. Another critical gene influencing milk composition is CSN2, which encodes beta-casein, a primary milk protein that significantly affects digestibility and consumer health. Beta-casein exists in several genetic variants, the most common being A1 and A2. The A2 variant has gained considerable attention due to its association with improved digestibility and reduced gastrointestinal discomfort in humans. Research conducted in New Zealand and published in Livestock Science demonstrated that cows producing A2 beta-casein milk exhibited no significant decrease in milk yield compared to cows carrying the A1 allele. However, there were subtle differences in casein micelle structure, influencing cheese yield and processing characteristics. Due to growing consumer demand for A2 milk, Genetic selection programs in countries like India, Australia, and the United States have focused on increasing the frequency of the A2 allele in dairy populations through selective breeding and genomic editing technologies. Lactose, the primary carbohydrate in milk, plays a fundamental role in determining milk volume due to its osmotic effects on water secretion in the mammary gland. The LALBA, or alpha-lactalbumin, gene encodes a key regulatory protein involved in lactose biosynthesis. Variants in LALBA have been linked to differences in milk yield, with certain alleles correlating with higher lactose concentrations and increased overall milk production. A study published in BMC Genomics examined the expression levels of LALBA in high-yielding versus low-yielding dairy cows and found that cows with elevated LALBA expression produced up to 12% more milk per lactation cycle. Nutritional interventions, particularly those involving energy-dense diets supplemented with glucogenic precursors, have been shown to upregulate LALBA gene expression, further enhancing milk synthesis. The ABCG2, or ATP binding cassette subfamily G member 2, gene is a well-established regulator of milk secretion and lactation persistence. This transporter protein is involved in lipid metabolism and plays a role in the efflux of bioactive molecules into milk. A functional SNP in ABCG2, specifically a quantitative trait locus at chromosome 6, has been linked to significant variations in milk yield. A study conducted on Holstein cattle and published in Animal Genetics found that cows carrying the beneficial allele of ABCG2 produced approximately 500 kilograms more milk per lactation cycle compared to non-carriers. Similarly, PRGC1A, or peroxisome proliferator activated receptor gamma coactivator 1-alpha, has emerged as a key player in regulating energy metabolism during lactation. This gene influences mitochondrial function and glucose utilization, directly impacting milk yield and metabolic efficiency. Research on transition cows has demonstrated that higher expression levels of PRGC1A are associated with improved lactation persistence and reduced risk of metabolic disorders such as ketosis. Feeding strategies incorporating dietary fatty acids, particularly omega-3 and omega-6 fatty acids, have been shown to modulate PPRGC1A expression, thereby optimizing milk production efficiency. Hormonal regulation of milk production is primarily governed by growth hormone and prolactin signaling pathways. Genetic variations in the DGAT1 gene, which encodes growth hormone, and its receptor have been linked to differences in milk yield and lactation duration. Studies in dairy cattle have identified single nucleotide polymorphisms in the growth hormone receptor that significantly affect somatotropic axis activity, leading to variations in milk synthesis. A landmark study in the Journal of Endocrinology analyzed the effects of growth hormone polymorphisms in Holstein and Jersey cows. It revealed that cows with the favorable growth hormone receptor variant produced 8% more milk per lactation cycle. These findings have prompted the development of genetic selection programs that prioritize high growth hormone responders to enhance lactation efficiency. Similarly, prolactin, a hormone crucial for mammary gland development and milk secretion, is influenced by genetic variants in the prolactin gene. High prolactin-producing cows exhibit prolonged lactation periods and greater milk persistency, making prolactin genotyping a valuable tool in dairy herd management. Beyond genetic variation, epigenetic modifications play a crucial role in regulating milk production genes. DNA methylation patterns in lactation-related genes such as STAT5A, FOXA1, and INSIG1 have been associated with variations in milk yield and composition. Studies have shown that higher methylation of STAT5A, a transcription factor essential for mammary gland development, correlates with reduced milk production in heat-stressed cows. Furthermore, histone modifications in milk protein genes have been implicated in lactation efficiency. Research in epigenetics and chromatin demonstrated that acetylation of H3K27 in the CSN2 locus enhances beta-casein expression, contributing to higher milk protein content. Nutritional strategies that modify epigenetic marks such as folic acid and choline supplementation have been explored to optimize milk yield at the molecular level. With the integration of high-throughput sequencing, bioinformatics, and genome editing technologies, dairy farmers now have access to sophisticated tools for genetic improvement. The use of genomic selection and polygenic risk scoring allows for more accurate prediction of milk production traits based on an animal's genetic profile. The application of CRISPR-Cas9 gene editing in dairy cattle holds immense potential for precision breeding. Scientists have successfully targeted genes such as FUT2, which is associated with milk oligosaccharides, to enhance milk's functional properties. While ethical and regulatory considerations remain, the future of dairy farming will likely be driven by a combination of genomics, nutrigenomics, and epigenetics to maximize productivity and sustainability. Epigenetics, the study of heritable changes in gene function that do not involve alterations in the DNA sequence, has emerged as a critical factor in understanding how diet influences milk production in dairy cows. Epigenetic mechanisms such as DNA methylation, histone modifications, and non-coding RNAs, particularly microRNAs, regulate gene expression in response to environmental and nutritional cues. These modifications can have profound effects on lactation efficiency, milk composition, and metabolic adaptation, shaping the productivity and sustainability of dairy farming. Nutritional interventions targeting these epigenetic pathways have demonstrated remarkable potential in optimizing milk production by modulating gene expression at the molecular level. DNA methylation is one of the most well-studied epigenetic modifications, involving the addition of a methyl group to cytosine residues in CpG dinucleotides, leading to gene silencing or activation depending on the genomic context. In dairy cows, the degree of methylation in key lactation genes has been found to significantly influence milk yield and composition. A study published in BMC Genomics analyzed the DNA methylation profiles of high and low-producing Holstein cows, revealing that high-yielding cows exhibited lower methylation levels in the STAT5A gene, a transcription factor essential for mammary gland development and milk synthesis. The reduced methylation allowed for greater STAT5A transcription, leading to enhanced expression of milk-related genes such as CSN2 or beta-casein and LALBA or alpha-lactalbumin. Conversely, cows experiencing heat stress or energy deficiencies exhibited hypermethylation in lactation genes, leading to suppressed milk production. Nutritional interventions aimed at modifying DNA methylation have shown promising results. Methyl donor nutrients such as folic acid, choline, methionine, and betaine play a crucial role in maintaining proper methylation patterns in lactation-related genes. A trial conducted at the University of Illinois found that Holstein cows supplemented with rumen-protected methionine exhibited increased milk protein and fat percentages, likely due to enhanced methylation-mediated gene regulation. Histone modifications, including acetylation, methylation, phosphorylation, and ubiquitination, influence chromatin structure and gene accessibility, ultimately determining whether genes are transcriptionally active or repressed. These modifications regulate key metabolic pathways involved in mammary gland function and energy utilization during lactation. Histone acetylation, mediated by histone acetyltransferases, or HATs, is particularly important in activating lactation genes. A study published in Epigenetics and Chromatin demonstrated that increased histone H3K27 acetylation at the CSN2 locus correlated with higher beta-casein production in dairy cows. This suggests that dietary strategies aimed at promoting histone acetylation could enhance milk protein synthesis. Nutrients such as butyrate, a short-chain fatty acid derived from ruminal fermentation of fiber, have been shown to act as histone deacetylase or HDAC inhibitors, promoting histone acetylation and increasing the transcription of genes involved in milk synthesis. Research at Wageningen University found that cows fed high-fiber diets rich in fermentable carbohydrates exhibited elevated milk yield and improved protein content, likely due to enhanced histone acetylation at key lactation genes. Histone methylation also plays a crucial role in metabolic adaptation during lactation. Trimethylation of H3K4, a histone mark associated with gene activation, has been observed in genes regulating lipid metabolism in high-producing dairy cows. Studies suggest that supplementation with polyphenol-rich feeds, such as grapeseed extract and green tea catechins, can modulate histone methylation patterns, improving milk fat composition by upregulating genes involved in fatty acid synthesis and desaturation. MicroRNAs or MicroRNAs are small non-coding RNAs that post-transcriptionally regulate gene expression by binding to target messenger RNAs or mRNAs and preventing their translation. In dairy cows, MicroRNAs play a crucial role in mammary gland development, lipid metabolism, and immune responses, making them essential mediators of nutrigenomic interventions. Recent studies have identified MIR33 and MIR223 as key regulators of lipid metabolism in dairy cows. A study in BMC molecular biology found that MIR33 targets SCD1 or stearoyl-CoA desaturase 1, a critical enzyme involved in monounsaturated fatty acid synthesis. Cows supplemented with omega-3 fatty acids exhibited downregulation of MIR33, leading to increased SCD1 expression and improved milk fat composition. Similarly, MIR148A has been shown to regulate milk protein synthesis by targeting mTOR, or mechanistic target of rapamycin, a key signaling pathway involved in protein metabolism. Dietary interventions that enhance amino acid availability, particularly leucine and methionine, have been found to suppress MIR148A, leading to increased mTOR activation and improved milk protein yield. Furthermore, exosomal miRNAs, secreted in milk and absorbed by offspring, play a role in programming metabolic traits in calves. Studies suggest that feeding high-energy diets during late gestation alters maternal miRNA profiles, influencing offspring growth and future milk production potential. This highlights the transgenerational effects of epigenetic modifications induced by nutrition. Given the growing evidence supporting the role of epigenetics in milk production, several nutrigenomic strategies have been proposed to optimize lactation performance through diet. Methyl donor supplementation, enhancing methylation capacity through folic acid, choline, and betaine supplementation has been shown to regulate milk synthesis genes and improve lactation efficiency. Butyrate and fiber-rich diets. Increasing dietary fiber intake promotes butyrate production, leading to histone acetylation and enhanced gene activation related to milk protein and fat synthesis. Polyphenol-rich feeds. Supplements containing grape polyphenols, green tea catechins, and flavonoids modulate histone methylation and lipid metabolism genes, improving milk fat composition. Omega-3. Fatty acids feeding flaxseed, fish oil, or algae-derived omega-3s downregulates MIR33, leading to increased monounsaturated fatty acid levels in milk. Leucine and methionine supplementation. These amino acids regulate mTOR signaling via MicroRNA interactions, promoting casein synthesis and higher milk protein yield. Early life nutritional programming. Feeding pregnant cows optimal diets influences exosomal miRNA profiles, leading to transgenerational improvements in calf growth and future milk production. Beyond diet, environmental stresses such as heat stress, pathogen exposure, and oxidative stress also influence epigenetic regulation of lactation genes. Heat-stressed cows exhibit increased DNA methylation of prolactin receptor, or PRLR, and STAT5A, leading to reduced milk yield. Strategies such as antioxidant supplementation, like vitamin E and selenium, and cooling systems help mitigate these epigenetic effects, ensuring consistent milk production during heat stress periods. Additionally, exposure to endocrine disruptors such as aflatoxins and pesticide residues can alter histone modifications and microRNA expression, negatively impacting mammary gland function. Feeding mycotoxin binders and organic selenium has been shown to counteract these epigenetic disruptions, safeguarding milk production. Milk fat composition is a critical determinant of dairy product quality, nutritional value, and market demand. Consumer preference has shifted towards dairy products rich in beneficial fatty acids such as conjugated linoleic acid, omega-3 fatty acids, and monounsaturated fats, while reducing saturated fats associated with cardiovascular risks. Nutrigenomics, which explores the interaction between diet and gene expression, provides a powerful approach to modifying the fatty acid profile of milk by targeting lipid metabolism at the genetic and epigenetic levels. By manipulating key genes, dietary components, and microbial interactions in the rumen, researchers and dairy farmers can optimize milk's nutritional profile, ensuring both economic benefits and health advantages. Milk fat synthesis is regulated by a complex network of genes encoding enzymes responsible for lipogenesis, fatty acid desaturation, and triglyceride assembly. The stearoyl-CoA desaturase 1 gene is one of the most extensively studied genetic regulators of milk fat composition. SCD1 catalyzes the conversion of saturated fatty acids such as stearic acid into monounsaturated fatty acids such as oleic acid. This process directly influences the proportion of saturated and unsaturated fats in milk. A study published in the Journal of Dairy Science analyzed SCD1 polymorphisms in Holstein cows and found that specific SCD1 variants were associated with a higher concentration of unsaturated fatty acids and lower levels of saturated fats in milk. Cows carrying the favorable A allele exhibited an 18% increase in oleic acid and a 12% increase in CLA, enhancing the health profile of dairy products. Moreover, dietary interventions that upregulate SCD1 expression have been found to improve milk fat quality without compromising total milk yield. Another key gene, FASN, regulates de novo fatty acid synthesis in the mammary gland. Research indicates that cows with higher FASN expression levels produce milk with increased levels of short and medium-chain fatty acids, which contribute to the creamy texture and flavor of dairy products. Nutrigenomic interventions targeting FASN activity through dietary lipids have successfully modulated milk fat composition. Nutritional strategies aimed at optimizing the balance of saturated and unsaturated fatty acids leverage specific feed additives and bioactive compounds that influence gene expression. These interventions target key pathways regulating lipogenesis, beta-oxidation, and lipid transport in mammary gland cells. Enhancing unsaturated fatty acids with omega-3 and omega-6 supplementation. Omega-3 and omega-6 fatty acids are essential polyunsaturated fatty acids that cannot be synthesized by the animal and must be obtained through diet. These fatty acids play a pivotal role in improving milk's lipid profile and enhancing the health benefits of dairy products. Feeding cows diets rich in flaxseed, fish oil, algae-based omega-3 supplements, and linseed oil has been shown to increase docosahexaenoic acid and eicosapentaenoic acid concentrations in milk. A large-scale trial at Cornell University found that cows supplemented with algae-derived DHA exhibited a 40% increase in omega-3 content in milk, while maintaining milk yield and quality. Another study in animal nutrition and metabolism demonstrated that feeding cows a diet containing 5% flaxseed for 8 weeks increased CLA levels by 35% and reduced the saturated to unsaturated fat ratio. The mechanism underlying this effect involves downregulation of SREPP1, a transcription factor involved in de novo fatty acid synthesis, leading to decreased saturated fat synthesis and increased PUFA incorporation into milk. Conjugated linoleic acid, or CLA, is a group of beneficial fatty acids with potential anti-carcinogenic, anti-inflammatory, and anti-obesity properties. CLA is produced in the rumen through microbial biohydrogenation of linoleic acid and also synthesized in the mammary gland by SCD1-mediated desaturation of vaccinic acid. Several dietary strategies have been employed to increase CLA levels in milk. Linoleic acid-rich diets, feeding cows diets high in sunflower seed, safflower oil and canola meal significantly increases CLA content in milk due to enhanced microbial conversion of linoleic acid in the rumen. Rumen-protected CLA supplements. Direct supplementation of rumen-protected CLA has been shown to elevate milk CLA levels by up to 80% without adverse effects on milk yield. SCD1 gene expression modulation. Studies indicate that omega-3 fatty acids and antioxidants such as selenium enhance SCD1 expression, leading to increased endogenous CLA synthesis. Saturated fats, particularly palmitic acid or C16-0 and myristic acid or C14-0, are abundant in milk and contribute to its structural integrity. However, excessive intake of saturated fats is linked to cardiovascular disease, prompting efforts to reduce their levels in dairy products, replacing palm fat with healthier lipids. Traditionally, palm-based fats are used in dairy cow diets to boost milk fat percentage. However, substituting palm fat with canola oil, soybean oil or fish oil has been shown to reduce saturated fat content by 15 to 25% while increasing unsaturated fatty acid. Targeting lipogenic enzymes, nutritional interventions that suppress ACC or acetyl-CoA carboxylase and FASN expression can limit saturated fat synthesis while promoting the incorporation of dietary unsaturated fatty acids into milk. The rumen microbiome plays a crucial role in regulating fatty acid metabolism by mediating the biohydrogenation of dietary lipids. Researchers have explored microbial-based nutrigenomic interventions to optimize milk fat composition, inhibiting rumen biohydrogenation. Feeding tannins, essential oils and ionophores such as monensin reduces excessive biohydrogenation of unsaturated fatty acids, leading to greater incorporation of healthier fatty acids into milk. Probiotic supplementation. Supplementing cows with probiotics containing lactobacillus and bifidobacterium species has been shown to modulate ruminal fermentation, increasing PUFA absorption and milk omega-3 content. Emerging research suggests that dietary components can influence DNA methylation and histone modifications, affecting genes involved in fatty acid metabolism. Histone acetylation and PUFA incorporation. A study in epigenetics and metabolism found that butyrate supplementation increases histone acetylation at lipid metabolism genes, enhancing the expression of SCD1 and FASN. Methyl donor effects on lipid genes. Feeding cows diets enriched with choline, folic acid and methionine has been shown to modulate DNA methylation patterns in lipid metabolism genes, leading to improved milk fat quality and composition. With the rise of precision dairy nutrition, the future of optimizing milk fatty acid composition lies in the integration of real-time genomic data, nutrigenomic modeling, and artificial intelligence-driven feeding programs. By using metabolomics and transcriptomics, dairy farms can customize feeding strategies based on individual cow genotypes, ensuring optimal milk fat composition while maintaining production efficiency. The integration of CRISPR-based gene editing also holds promise for targeting lipid metabolism genes to enhance milk fat composition without requiring drastic dietary changes. Research in genetic engineering of SCD1, ACACA, and PPRGC1A could revolutionize dairy production by producing milk naturally enriched with health-promoting fatty acids. The transition period, spanning 3 weeks before and after calving, is a critical phase in dairy production where cows experience drastic metabolic shifts. Negative energy balance during this period can impair milk production, compromise immune function, and lead to metabolic disorders such as ketosis and fatty liver syndrome. Nutrigenomic strategies aimed at optimizing energy metabolism have been instrumental in mitigating these challenges. Research has identified PPRGC1A, a gene involved in mitochondrial biogenesis and energy metabolism, as a key target for nutritional interventions. Studies have shown that supplementation with rumen-protected choline enhances PPRGC1A expression, promoting efficient lipid mobilization and reducing the risk of metabolic disorders. Similarly, dietary incorporation of niacin and chromium has been found to modulate insulin sensitivity, thereby improving glucose utilization and milk yield. Furthermore, proteomic analyses have revealed that cows experiencing negative energy balance exhibit downregulation of mammary gland proteins involved in milk synthesis. Nutritional strategies such as bypass protein supplementation and rumen-protected amino acids have been shown to counteract these effects by upregulating milk protein synthesis genes. Such findings reinforce the significance of precision nutrition in maintaining lactation performance during metabolic transitions. Beyond milk production, nutrigenomic interventions play a crucial role in addressing the environmental impact of dairy farming. Methane emissions from enteric fermentation contribute significantly to greenhouse gas accumulation. Researchers have explored dietary modifications that suppress methanogenesis while maintaining rumen health. The MTR and MMMUT genes involved in methanogenic pathways have been identified as potential targets for reducing methane output. Studies have demonstrated that dietary supplementation with tannins, essential oils, and nitrate-based compounds can downregulate these genes, leading to lower methane emissions. A large-scale study published in Animal Feed Science and Technology found that feeding cows 3-nitrooxypropanol reduced methane emissions by 30% without compromising milk yield. Additionally, integrating seaweed-based feed additives has shown promising results in methane mitigation. A recent experiment conducted in Australia reported a 60% reduction in enteric methane production when cows were supplemented with Asparagopsis taxiformis, a red seaweed known for its anti-methanogenic properties. Such advancements underscore the potential of nutrigenomics in enhancing both productivity and environmental sustainability in the dairy industry. While nutrigenomics presents immense potential in optimizing milk production, several challenges must be addressed to ensure widespread adoption. The high cost of genomic testing and precision feeding strategies remains a barrier for small-scale dairy farms. Additionally, individual variations in gene-diet interactions necessitate personalized nutrition approaches, requiring further refinement of predictive models. Ethical considerations surrounding genetic modifications and dietary interventions must also be carefully navigated. Regulatory frameworks governing nutrigenomic applications in livestock production must balance innovation with animal welfare and consumer safety. Moreover, long-term studies assessing the sustainability and economic feasibility of nutrigenomic strategies are essential to validate their efficacy. Despite these challenges, the future of nutrigenomics in dairy science appears promising. The integration of big data analytics, machine learning, and artificial intelligence in precision feeding programs will revolutionize the industry, enabling real-time optimization of milk production at the genetic level. As research continues to unravel the molecular intricacies of lactation, nutrigenomic interventions will play an increasingly pivotal role in shaping the future of sustainable and efficient dairy farming. Nutrigenomics represents a paradigm shift in dairy science, bridging the gap between genetics and nutrition to enhance milk production and quality. By harnessing the power of gene-diet interactions, researchers have developed targeted nutritional interventions that optimize lactation efficiency, improve milk composition, and mitigate environmental impact. The integration of genomic selection, epigenetic modulation, and precision feeding strategies offers a sustainable pathway to revolutionize the dairy industry. As technology advances and scientific understanding deepens, nutrigenomics will undoubtedly remain at the forefront of dairy innovation, shaping a future where milk production is optimized at the molecular level for both economic and environmental benefit.